pleasure to introduce our second speaker. Um, Matthew, I think, okay, great. Yeah. So our second speaker, Matthew, who is a group leader and a well-contrast CDA fellow at the Wellcome Center for Cell Biology, where his group used quantitative approaches in cell as well as molecular biology to understand how different cellular processes are rewired as the cells grow and they change their sizes. So after studying natural science at the University of Cambridge, Matthew obtained his PhD in Sir Paul Nurse's lab at the Francis Crick Institute, working on cycling CDK substrate phosphorylation dynamics. In 2017, he moved to Stanford as a postdoc with Jens Gosian, where he used the quantitative functional genomics approach to address the long-standing question about how global transcriptional output is scaled with cell size. He then moved to Edinburgh to establish his own group at Well Contrast Center for Cell Biology in 2023. So today he's going to tell us about RNA pol-2 dynamics and mRNA stability feedback scale mRNA amounts with cell size. Thank you, Matthew, for coming, and the floor is yours.
Fantastic. Thank you very much. Can you hear me okay?
Yeah,
Yes?
we can hear.
Okay, great. All right, well, thank you for the kind introduction and the invitation to speak. As Sky said, I'm going to talk today about a piece of work that I did as a postdoc in Jan Schottheim's group, trying to understand this long-standing question about how RNA amounts are coordinated with changes in cell size. So my interest and my lab's interest is in how different processes inside the cell are coordinated with changes in cell size. And cell size is a fundamental property of all cells. Of course, all cells have a size, and size also changes across a range of different biological contexts. And even if you think about a single proliferating cell, it has to change in size approximately twofold between when it's born small and when it grows to a larger size before it divides. And so that's kind of schematized here by these cartoons of budding yeast, which are our model system of choice for the work that I'm going to show you today. So as cells do this, as they change and change their size during the process of growth, they have to coordinate different aspects of their cell biology with these changes in cell size. And maybe the most intuitive example of this is the scaling of intracellular organelles. So the volume of organelles, such as the nucleus, increase approximately in proportion to cell size, so they occupy a fixed fraction of cell size. But it's not just the internal structures of the cell that are coordinated with cell size, but also the total amount of macromolecules. So the total amount of RNA and the total amount of protein inside cells has to increase continuously as the cell grows and increases in volume, despite the fact that the template for RNA synthesis, the genome, of course, increases in a discrete and discontinuous manner during the cell cycle as cells are growing. Okay, so this scaling of macromolecules with cell size is something that was really first appreciated back in the 1970s from really classic radio labeling experiments. But there's been kind of several more modern approaches, more recent studies using a combination of genomics and single cell imaging to kind of revalidate and reconfirm this observation of scaling of different macromolecules. And the reason that this scaling of macromolecules is so important is it ensures something that we call concentration homeostasis. So the concentration of any given macromolecule is just a function of its copy number over its volume. So by scaling copy numbers in proportion to cell size, concentrations are kept constant. This ensures that, of course, concentrations of enzymes and reactants specify reaction rates inside the cell. So by maintaining concentrations, biochemical reactions are sustained, and this underpins the functionality of all core cellular processes. And the question that I want to talk about today is how is this achieved, and how is it achieved specifically in the case of mRNA? And the reason this has been such a conundrum is something that I alluded to already, which is that as cells grow during one given stage of the cell cycle, the amount of DNA inside the cell is approximately constant. And the concentration of proteins, including all the transcriptional machinery, is also constant because of this scaling of protein amounts. So superficially, nothing is changing upstream of transcription, but then somehow the production of mRNA is changing and being coordinated with cell size. So to start to address this question, the first thing that we did was to look directly at RNA pol-2, the enzyme synthesizing mRNA, and measure how much of it is actively engaged on the genome. So we took a panel of different cell size mutants in budding yeast, And then we use this spike in normalized version of ChIP-seq that allows us to resolve global differences in occupancy that are not kind of typically afforded by conventional ChIP-seq to look at RNA polymerase occupancy genome-wide. And so looking at cells of different sizes, we can see this nice kind of continuous and gradual increase in the amount of total RNA pol that's engaged on the genome. That's not just true of polymerase across the whole genome, it's also true of polymerase inside gene bodies. You can see here in these metagene profiles. It's also not true of just the total population of RNA pol, it's also true of the initiated and the elongating versions of RNA pol that can of course be distinguished with different phosphoantibodies. Okay, so this was looking at cells of different sizes using different size mutants. We could also manipulate size by collecting very tiny cells and then arresting them in the cell cycle to allow their size to increase, and then performing the same experiments where we also reproduce and observe this scaling of RNA polymerase engagement on the genome. So this is probably not a huge surprise given what I said in the first place. I told you that there was more mRNA in larger cells, and here I'm showing you there's more transcriptionally active RNA pol2. So really the question becomes, how is it possible that larger cells load more RNA polymerase onto their genome to result in more transcription per unit time, per unit gene? And the main hypothesis that has been proposed and discussed in the literature over the last decade and a half has been that there could be some potential limiting component to the transcriptional machinery. And if there was a limiting factor whose could be responsible for coupling cell size to the global transcription rate under certain biochemical regimes, it may be sufficient to explain this kind of broad global scaling phenomenon. I kind of left that intentionally vague because we're going to get into a bit more kind of specifics about how exactly that works. But at this point, the focus that we then had at this stage was to say, well, if this is the case, we should be able to identify some limiting component to the transcriptional machinery, some dosage limiting component. Okay, so to test which bits of the general transcriptional machinery for mRNA synthesis are dosage limiting, we set up the following experiment. We tagged a subunit of each of the different subcomplexes of the pre-initiation complex with this FRB domain, which allows you to conditionally sequester it into the cytoplasm upon the addition of rapamycin. This is an approach called anchor away, but critically we set it up in a way where we only tagged one of the two alleles with this FRB domain. So in that way we can conditionally deplete half of any given factor from the nucleus in a short 30 to 40 minute window. And when we did that, we were somewhat surprised to find that actually most of the basal transcription factors, the general transcriptional machinery, do not really have any meaningful impact on the amount of transcription happening inside gene bodies when we deplete half of their amounts out of the nucleus. And that's all of these in these kind of blue, green, teal colors. You know, there's some effect here with TBP, there's no effect up here with TF2B, and in between there's a range of very modest effects. The one exception to this is RNA polymerase II itself. So a 50% reduction in the amount of RNA pol2 inside the nucleus results in about a 40% reduction in the amount of transcriptionally active RNA polymerase engaged inside gene bodies. And so this suggests that polymerase amounts and concentrations inside the nucleus are limiting for its own recruitment and initiation. And if this is the case, then we should be able to do the converse experiment. We should be able to overexpress RNA pol inside cells and ramp up the global transcription rate in a conditional way. So we set about to do exactly that experiment. We constructed all the 12 subunits of RNA polymerase II under separate tetracycline and G-spore promoters. We integrated all of them together into a single strain in addition to the endogenous copies of the polymerase subunits. And then upon addition of tetracycline, we get about a 30 to 40% increase in the amount of nuclear RNA polymerase after a short kind of 30 to 40 minute pulse, and we get a 30% increase in the amount of RNA pol that we can quantify inside gene bodies. So this really reinforces and kind of re-emphasizes this idea that polymerase is itself limiting for its own recruitment and initiation, because we can overexpress or partially deplete it from the nucleus in a conditional way and retune the global transcription rate. Okay, so with that, we kind of circle back to the initial question, and you know, the next thing to identify is really we have this limiting component now, and we can say that pol limits its own recruitment, but what has that got to do with the scaling phenomenon? Because these experiments that I've just been showing you, we've been messing around with RNA polymerase concentrations, but total RNA pol concentrations inside the cell don't change because the amounts of RNA pol scale in proportion to the cell size, like all other proteins, so its concentrations are approximately constant. And the reason that this scaling, this limiting factor may relate to scaling is of course because what really matters inside the cell to initiate transcription is the amount of RNA pol that's inside the nucleoplasm. And so to get a quantitative understanding about how limiting polymerase can result in this transcriptional scaling phenomenon, we formalized and compared a few different models, mathematical models, for how limiting polymerase could result in a transcriptional increase. So this is the model that we kind of ended up converging on and proposing, which we call a dynamic equilibrium model. This basically treats RNA polymerase as a population of free and bound RNA pol, which when added together, as I just showed you, are proportional to cell size. And then we can say that the rate at which polymerase exchanges on the genome is just a function of an on rate, the free populations of RNA pol times the amount of DNA, minus an off rate times the bound population. So this is very simple mass action kinetics. We can say this is set in equilibrium. We can then solve these equations, take the solution, which has just two free parameters, which are effectively the RNA pol concentrations inside the cell and the KD for it exchanging on the genome. And then we can take those solutions and fit them to the data on the left-hand side. And so when we do that, we get a pretty reasonable fit, which is reassuring. But at the same time, we also considered an alternative model that had also been proposed and discussed in the literature. And that model was also required polymerase to be limiting, but it treated polymerase as something with an extremely high on rate so that it was effectively titrated against the genome, so that all the polymerase inside the cell is bound to the genome, so that the total amount is proportional to the bound amount, so that you get some initial directly proportional and linear increase proportional to the amount of pol. And then at some point you would hit a saturation point and no more polymerase could be loaded. So this titration saturation model that's been discussed by several bits of theoretical work also does a pretty reasonable job of fitting to the data we have from our ChIP-seq experiments. And so of course what we wanted to do was to take these two models and start to compare and contrast what different predictions they make, and then go about experimentally testing to delineate and distinguish between these two models. So the the first kind of important prediction that these models make that allows us to distinguish them is what the bound fractions of RNA polymerase should do. So the fitting of this dynamic equilibrium model to our data specifically predicts, is based on fitting to the ChIP data, which is just a relative fold change in how much RNA pol is bound to the genome, but doesn't say anything about the relative ratio between bound and unbound fractions. But the fitting of our model to that data says that the bound fractions of RNA polymerase should start out at about 50%, and that should decline gradually and continuously as cells grow and increase in size. By contrast, this titration saturation model, by definition, says that all the RNA pol should be engaged on the genome until you hit a saturation point, and then you should get this kind of decrease in the bound fraction, which is just proportionate to 1 over volume. And so to test this directly, we teamed up with Rodrigo Reyes at McGill University, who's a real expert in single molecule tracking, to track individual molecules of RNA polymerase in cells of different sizes. So we're using a halo tag, we label the halo tag with a dye, and then we can use that to monitor its diffusive properties inside the nucleoplasm, and then distinguish the bound and the unbound populations of polymerase. So here there are some control molecules you can see that are kind of nicely bound histones, and then a free fluorophore that's floating around inside the nucleoplasm. And then for RNA pol, we have a mixture of these two things. So for RNA pol, what we then did is we measured dozens of molecules in hundreds of cells, and then for each cell we can classify what fraction of polymerase is bound. We can then bin cells according to their size, and then plot those bins on top of the data on the left-hand side. And so that's what I'm showing you here. This is our empirical measurements of bound fractions of RNA pol. in G1 cells as a function of cell size. And what we see is that the bound fractions are clearly very divergent from this titration saturation model, ruling that out, but do a reasonable job of sitting within the boundaries of the model predictions for this dynamic equilibrium model. Okay, so we wanted to push this a bit further and test a few other predictions or requirements of this model, this dynamic equilibrium model for size-dependent transcriptional scaling. One of the, I'm going to talk about two in the next couple of slides. The first is that this model basically assumes that the K-on for polymerase is effectively constant as a function of cell size, because the increase in loading that you get is really driven by changes in the concentrations of the free population of polymerase inside the nucleoplasm. And another way of saying that is that the upstream chromatin environment into which RNA polymerase is recruited should not change significantly as a function of cell size. So we set about doing two different types of experiments to test exactly that. The first was to use this single molecule footprinting assay where we treat nuclei with a cocktail of methyltransferases to measure global chromatin accessibility read out by DNA methylation. And so when we do this in cells of different sizes using a spike in for normalization, what we see is there's really very modest, almost no real meaningful changes in global chromatin accessibility over this kind of wide range in sizes. The other thing that we did is we took the same samples that we'd done a series of ChIP-seqs on for RNA polymerase, and we started to ChIP-seq a panel of different histone modifications. And so that's what I'm showing you here. And so we can then, as the same as the RNA poll, compare the relative change in the histone modification as a function of cell size. There are two examples here, one that's flat and one that's increasing. So we can kind of classify based on these slopes whether they change in a size-dependent manner. And the vast majority of histone modifications that we looked at did not change as a function of cell size. And the four that we have here that are exceptions to this, we can kind of ascribe as being downstream of RNA polymerase. So they do not necessarily represent the upstream environment into which RNA poly is recruited, but are deposited in a way that should be dependent on the upstream recruitment of RNA poly. And so this kind of summary graph, you can see the same results here in these heat maps displaying the size-dependent changes in all these different histone modifications on the right-hand side. Okay, so with that, there's then one final prediction of this model that for us was really the killer experiment, and that's this model also predicts the global transcription rate should be changing in a way that depends not just on size but also on DNA content. But it doesn't just predict that a change in the total amount of DNA should change transcription. It makes a very specific quantitative prediction about how that change should look and exactly how much more RNA pol should be engaged on the genome in a diploid compared to a haploid. And so what's here on the left is exactly that. In green is the quantitative prediction from this model of what happens in a diploid based on fitting the model to the haploid data. So we just took the solution and the fit in the haploid data and we stick a 2 in front of the DNA term, and then you get this curve. So we went away to do exactly this experiment and compare our haploid data to a diploid experiment. And so here you can see we have G1 arrested haploid cells compared to G1 arrested diploid cells. When we do that, we get a really nice tight convergence between our empirical measurements and the model prediction. And so this, with some of the preceding data and other experiments that I haven't had a chance to show you, started to make us think that we should take actually quite seriously this model as a mechanistic and quantitative framework for understanding how RNA polymerase recruitment, it's limiting RNA polymerase through the simple mass action recruitment kinetics onto the genome, can explain the size-dependent increases in RNA polymerase that we've been measuring. And so, at some point we were kind of feeling reasonably good about ourselves that we made some progress in understanding this problem, and then we realized it actually threw up a fairly significant problem for us, both this model and our data. And that was the fact that both within these equations and our empirical measurements, the increase in RNA polymerase occupancy in the genome is actually not particularly close to being proportional to cell size. So I started this talk by saying RNA amounts need to increase approximately in proportion to cell size to stay at a constant concentration, but that is not what we see comparing either the cell size mutants or our G1 arrest experiments when we look at the scaling of polymerase occupancy. Here we have a fold range of about almost 5-fold increase in cell size, and we get, at best a 2-fold increase in the amount of RNA pol that's engaged on the genome. And so if the scaling was perfectly proportional, it would fall on this dashed gray line. And so, of course, what we did in these situations is we asked, well, in addition to the transcription rate, we then used what happens to mRNA amounts. And so we used a spike-in normalized version of mRNA-seq to measure the total amount of mRNA in each of these different conditions. And what we see in both sets of experiments is that the total amount of RNA per cell increases more and far closer to being proportional than the total transcription rate. And in this initial regime, the amount of RNA pol inside the cell is pretty close to being proportional, and it does ultimately deviate, but there is a significant gap between the total amount of mRNA inside cells and the RNA polymerase occupancy. And so if we take both these sets of measurements, the mRNA-seq and the ChIP-seq at face value, there's only really one prediction that you can make from these, and that's that there should also be a global adjustment in mRNA decay rates as cells get larger. And you can quantitatively predict exactly what that change in decay rate should be just by comparing the difference between these two curves. So we wanted to then go and set up an experimental framework to test exactly that. So we use this EU pulse chase workflow where you label cells with a uridine analog called 5EU. That incorporates into newly synthesized RNA. We can then wash out the EU and follow it with a chase of high concentrations of kind of normal uridine. And so the EU labeled fraction will be being degraded and being replaced by the synthesis of unlabeled mRNA. And so we follow the chase with a time course, and then we perform an enrichment on the EU fraction and an enrichment for the mRNA fraction, and then sequence them with spike-ins to measure the ratio between the EU fraction and the unlabeled fraction. And so when we do that, we can measure decay rates quite nicely. This is the conditions where we have small cells at the beginning of the experiment, at the beginning of an arrest. And from this, we can fit an exponential nicely and calculate a decay rate or a half-life. And then at the same time, we can do a comparison of what should happen when we jump up about 3-fold in size. And from the graph that I showed you before, our model makes a very specific prediction. There should be slightly less than a 2-fold reduction in mRNA decay rates comparing these two specific conditions. And so when we do exactly that experiment, you can see that globally mRNA degradation rates have slowed down. The EU label fraction declines slower in these larger cells compared to smaller cells, and we get a very close empirical measurement, a very close agreement between our empirical reduction in global mRNA decay rates and the model prediction. It's not just the total amount of EU. This isn't driven by some subfraction of particularly highly expressed transcripts. It's also true of transcripts across the entire transcriptome. And so this is just shown here as the cumulative distribution of the half-lives of all the transcripts for which we could fit and calculate decay rates for the small cells in gray and the larger cells in blue. Okay, so that more or less comes to the end of the data that I wanted to present and talk to you about today. I'm just going to quickly summarize and say that what we've been able to show is that as cells get larger, more transcriptional machinery is loaded onto the genome in larger cells. And that we've been able to identify that unlike other components of the basal transcriptional machinery, RNA pol2 is the major dosage limiting component of the pre-initiation complex. And we can understand how limiting polymerase results in a size-dependent increase in transcription rates through a very simple mass action model that treats polymerase as being in a kind of a simple dynamic equilibrium exchanging on and off the genome. But this is also in a context where the upstream chromatin environment, both in terms of upstream histone modifications and chromatin accessibility, is largely invariant as a function of cell size. at least within the range of sizes that we're examining and probing here. But this global increase in transcription rates that we quantify by itself is not sufficient to explain the scaling in mRNA amounts that we see. It's not enough to keep RNA concentrations close to being constant. And there is, in fact, some additional feedback mechanism operating on mRNA stability that helps stabilize the transcriptome in larger cells and keep RNA amounts closer to being proportional to cell size than they would be if it was left to transcription alone. Okay, so all of this stuff is now published at the end of last year. I will quickly say thank you to Jan, whose lab I did all this work in, the people in Jan's lab who contributed to the project, as well as Georgi at Stanford, who was really instrumental in a lot of the bioinformatics, and Juan, who did all the single molecule tracking experiments that I showed you, as well as colleagues at the CRIC who contributed to some experiments that I didn't have a chance to introduce. Of course, to funding bodies, and I'm also very grateful to have now recently moved to Edinburgh and have started to set up my lab here. And we have several postdoc positions available. If people are interested, then please get in touch. I'd be very happy to talk to you. So with that, I will conclude, and I'll of course be very happy to take any questions.
Thanks, Matthew, for the great talk. And if you have any questions, please either type them in Q&A box or raise your hands so we can allow you to ask yourself. So the first question you got, it's a fantastic talk and the cell paper. Have you considered how cell size might impact transcription elongation rate? Slower elongation rate could increase residence time over each gene and therefore tripability of PAL2 without necessarily increasing initiation rate. Does DNTP concentration change with scale size?
So we don't know about nucleotide concentrations. I think the elongation rates is a really interesting thing. We don't see any evidence in our data for changes in elongation rates as a function of cell size. But one thing that we've started to think about is whether, the global transcription rates also change in different environmental conditions. And so if you were to take our model at face value without changing elongation rates, you could imagine that as cells switch into a poorer nutrient condition, to reduce global transcription rates, you would have to have a far lower bound fraction, and that would then mean this equilibrium model wouldn't really work and you wouldn't get a nice scaling relationship. But if elongation rates also slowed down to kind of be coordinated with any reduction in initiation, then this size-dependent scaling could be maintained across different environmental conditions, different growth media or nutrient conditions, for example. So that's one thing that we're super interested in looking at, whether as you vary environmental conditions and you want to tune your global transcription rate, not just in response to cell size, whether the elongation rate can help you do that whilst also keeping that size-dependent transcription in any given condition. Hopefully that makes sense and answers the question.
Okay, thank you. The next question is, how does the fraction bound of general transcription factors scale with cell volume?
So we didn't get into enough measurements we didn't do enough single molecule measurements on them to say too precisely exactly what's happening. My feeling, we did some proteomics, which is not an estimate of the bound fractions, that's just an estimate of the relative change in how much is on the genome. And we see all the general transcription factors increasing, but also those increases are dependent on polymerase recruitment. So if we deplete RNA pol, then we see a loss of their association. But starting to look at all the basal transcription factors with the experiments to look at proper bound fractions with the single molecule imaging would also be super nice, and would love to start getting into some of that. What I think is known though in general is that the bound fractions of most of the basal transcription factors does tend to be lower than RNA polymerase itself, and it kind of varies for them, but I think in yeast it tends to be more in the kind of 10 to 30% range, which is consistent with the idea that there's more of them available and they're less likely to be the thing that's immediately limiting at any given cell size.
So the next question is it only the PAL2 affected by this scaling? How about other polymerases? Do they follow the same models to scale up other molecules?
Yeah, and we'd like to look at exactly that. That's one of the things that we're planning to start to look into now. We focused on pol 2 because I guess you've got to start somewhere, and pol 2 is not an unimportant polymerase. But what we do know is the M, mRNA amounts and rRNA amounts track each other. So we've done like total RNA-seq where we can compare the ratio between those two things. So the scaling at the transcript level seems to be coupled. There are coupling processes, between RNA pol 2 and pol 1. So we'd like to get into that and basically do some of the same experiments that I've shown today, but are looking first at pol 1 and then probably after that at pol 3. But yeah, a great question.
So what happened to the number of ribosome or global translation rates as the function of size or decreased mRNA decay?
That we don't know, but we are also trying to measure exactly those things at the moment. Part of the reason for this is that as the mRNA decay rates change, there's two reasons that we're kind of interested in looking at it. One is that you could imagine there is a known competition between translational machinery and mRNA decay machinery. So it's possible that changes in the translational machinery could actually be responsible for this change in mRNA decay rates. And then conversely, probably kind of apparent from our data, hopefully, that even though the mRNA amounts scale more than the transcription rate, they do eventually not scale perfectly, right? So the scaling is still imprecise and imperfect. And so, we'd like to understand how that then impacts translation rates, whether mRNA becomes limiting for translation at that regime, or whether ribosomes are still the limiting component and translation rates do not change. So those are all things which I wish I knew the answer to, but we are like actively trying to identify and quantify at the moment.
So the next question is, since the DNA content remains the constant with cell size, what causes the change in pol2 occupancy? Does the genome become more accessible with size, or does the epigenetic changes occur with cell size?
So it's none of those things. The chromatin accessibility, like we measure, doesn't change. The upstream epigenetic modifications, histone modifications, do not change. What drives it is really the simple mass action kinetics of RNA pol. So I can give maybe an intuitive sense. If you imagine a cell doubling in size instantaneously, has the same amount of DNA, but then there will be more RNA polymerase produced. In the first instance, because RNA polymerase amounts are proportionate to size, in the first instance, that polymerase would enter the free fraction, right? So that would significantly increase the free concentration, even though the total cellular concentration is not changing, which would push more of it onto the genome until you establish a new equilibrium between the bound and unbound populations. So this increase really emerges from like one simple fact, which is that polymerase is limiting, and that it's in an equilibrium where about half of it is bound and half of it is not bound. And that by itself is, both sufficient to explain the scaling we observe, and then, verified by several different measurements, orthogonal measurements we made with the single molecule tracking and the ploidy comparisons. But that's not to say that, because of the ploidy comparisons, we can say that in addition to polymerase amounts, DNA content also instructs the global transcription rate, which may provide a bit of a window into why different tissues, as they get larger, often undergo polyploidy. So they may have some coordinated connection between DNA content and cell size in different physiological situations.
So we still got a lot of questions. Okay. Does the pol2 and the transcription rate changes in similar way in cells that are polynucleated?
I would assume so. We haven't measured that. I mean, one of the things that all of this, we hope to understand how all of this ends up connecting to very basic properties like growth and translations we were kind of discussing a moment ago. And what is known is if you have a haploid cell that gets larger, you have, as we showed, this sublinear scaling of transcription, and ultimately there is a decline in growth rates. That isn't true when you increase your size, but you also increase your DNA content in a single nucleus. So DNA can allow transcription rates to increase more than polymerase alone. So what I would suspect looking in multinucleated cells is it would behave the same as a polyploid cell, even where the ploidy is in a single nucleus. So the DNA content is a really important parameter in this, just as much as the RNA pol. And I think that kind of goes back to something I mentioned in the response to the last question. There are lots of different cell types in your body where the largest cell types in your bodies are the ones with kind of significant polyploidy, and that may be exactly why that's happening.
Can you comment on the nuclear depletion of some of the PIC members? Why did you think there's no changes in polymerase occupancy?
So we're not measuring we're not depleting all of them, right? So we're doing an acute 50% reduction. So we remove half of them out of the nucleus and we don't see much of an effect. So the basic way of interpreting that is that they are, of course, important for transcription, essential for transcription, but their nuclear concentrations, when you change them twofold, are significantly high enough that it doesn't alter or impact the RNA polymerase recruitment kinetics. Obviously, there's buckets of data over the last couple of decades showing when you deplete these things, transcription stops. But we're not removing all of them. We're only depleting half of them from the nucleus.
The next question is, did you think mRNA concentration might scale more perfectly with cytoplasmic volume as opposed to total cell volume, which might include the translational irrelevant compartments of the nucleus?
So, at least with regards to we think that the cytoplasmic volume is pretty closely proportional to the cell volume. I mean, the nucleus, for example, we know is directly proportional. So the fraction that is the cell minus the nucleus is also directly proportional to cell size. In budding yeast, at some point when cells get very large, the vacuole can take up a significant fraction of cell volume. And that may increase, but I think that's well beyond the size ranges we're looking at. That's when cells start to get quite sick, then the vacuole starts to increase it with respect to other organelles. But most organelles scale pretty closely in proportion to cell volume, meaning that everything else in the cytoplasm is also proportional to the cell volumes that we measure.
So I think you kind of answered the next question, which is how does the ratio of the volume of the nucleus to the total volume of the cell affect this?
Yeah, and so that's that's an important parameter in the model that it has to be, roughly proportional. But there are now lots of kind of really nice data across dozens, well not dozens, across like a nice spread of different organisms showing that as cell size increases, nuclear volume increases. The data that I showed at the beginning of my talk was maybe the most kind of beautiful of that data, which is work from Paul Nurse's lab in fission yeast that shows this really wide kind of perfectly proportional scaling. We made all those measurements as well, and they're in the supplement of the paper, and we see pretty close to proportional scaling. It's not perfectly proportional. There's some little deviation, and that actually goes into our model, but it doesn't really make a difference. If we assume it's perfectly proportional, or we put into the model our empirical measurements, the model does exactly the same thing.
So maybe last question, with scaling, it's the change of transcription initiation uniform across all loci, or there's a preference or bias in terms of relative usage of initiation sites?
Yeah, so one thing that it's an excellent question. That's also something we're interested in getting into. It's definitely clear that across a range of sizes, there are some genes that increase their transcription rate more than other genes, more than the average gene, and some that decrease their transcription more than the average gene. And so in the cell size field, those have been kind of termed superscaling and subscaling genes respectively. There are different classes of transcripts that seem to obey those properties. We know, for example, that the histones do not scale their production with cell size. So the synthesis of histone transcripts is completely flat when you have a fixed amount of DNA, which of course makes sense. A cell produces histones in proportion to its genome rather than in proportion to the volume of the cell or the volume of the nucleus. There are other examples which have been kind of fairly carefully examined, including cell cycle inhibitors. So as cells get larger, they produce less cell cycle inhibitors, and that actually allows natural cells to re-enter the cell cycle faster because you reduce the concentration of cell cycle inhibitors when cells get too big. But mechanistically, how those things are happening is not fully clear. one proposal is that some genes may actually start to saturate when, in normal conditions, if they're close to saturating, if you then try and increase transcription, they're not able to increase it anymore. That might make sense in the context of the histones, which are, when they're on, very highly expressed. But there are lots of transcripts that have sub or superscaling behaviors, where the initial occupancy does not really seem to have, I would say, adequate explanatory power. So I think that happens. Why, we don't really understand, or we can kind of imagine why the cell is doing that in terms of its functional outputs, but how it's doing that, we still don't really understand, and that's certainly part of the next trench of questions that we need to get into.
Thank you. Thank you, Matthew, for answering all the questions. I think that would be today's session. Thank you for both speakers for the wonderful talk and all the wonderful questions from the audience. It's a really, really a lot of discussions. Yes, thank you.
Ben has one question.
Oh, okay. Sorry, Ben, I forgot to see your raised hand.
Oh, no, that's fine. Just wonderful talk, Matthew. And you can see that by the very engaged Q&A. I had one question, which was about squelching and mediator. So there's that old squelching phenotype, which they helped to find mediator. In the end, I was wondering, in your cell size scaling, did you ever look at mediator? And do you think that also may be limiting?
We didn't, partly because we wanted to do these partial depletions. And the problem with mediator in yeast is there are so many different components to it, they don't come all out together. So you have to start tagging multiple ones. And the second you do that, you don't really know what on earth is going on with your experiment, because we care about, what's dosage limiting rather than what's essential. So we wanted to do that and include that along the other basal transcription factors we looked at, but we didn't get into it. We do have some chromatin proteomics, like an approach called CHEP, where we purified chromatin and then just subjected it to mass spec, where we see all the basal transcription factors as well as RNA polymerase increasing, and mediator does the same thing. So it kind of follows the trend. And I guess other than that, the only thing I can say is that we don't need to think about mediator to, I guess, explain the observations we have. Polymerase by itself has been sufficient, but that doesn't mean that it doesn't play a role. I could imagine a more complicated version of the simple model I proposed, but that was also true.
Awesome. Thank you.
Thank you. I think that's the official end.
All right. Fantastic.
Thank you all for attending. Yeah. Thank you for the wonderful talk. Yeah.